Chemical bonds play a crucial role in determining the properties and behavior of molecules. They arise from the interactions between atoms, which can be classified into two broad categories: strong and weak interactions. The strong chemical bonds or interactions that can be disrupted by more energy, and the other is weak chemical bonds or interactions that can be broken by weak forces like thermal motion. The strong interaction. Covalent bonds. One of the most fundamental and prevalent types of strong chemical bonds is the covalent bond. Covalent bonds occur when atoms share electrons in order to achieve a more stable electron configuration. This sharing of electrons allows atoms to complete their valence shells, resulting in a more energetically favorable state. Covalent bonds are characterized by the equal or unequal sharing of electrons between atoms, depending on their electronegativities. In a pure covalent bond, such as in diatomic molecules like oxygen, O2, or nitrogen, N2, the shared electrons are equally attracted to both atoms, creating a balanced distribution of charge. This type of bond is nonpolar. On the other hand, in polar covalent bonds, such as in molecules like water, H2O, or ammonia, NH3, the electronegativity difference between atoms leads to an unequal sharing of electrons. This imbalance results in partial charges on the atoms, making the bond polar. Covalent bonds are incredibly strong and require substantial energy input to break. They contribute to the stability of molecules and are responsible for a wide range of chemical properties, including bond lengths, bond angles, and molecular geometries. Covalent bonding allows for the formation of intricate and diverse molecular structures, enabling the complexity of organic compounds and the richness of biological systems. The strong interaction in biology. Chemical bonds are essential in the realm of biology as they dictate the structure, function, and behavior of biological molecules. From the intricate folding of proteins to the recognition of key molecules in cellular processes, understanding the strong and weak interactions of chemical bonds is crucial in unraveling the mysteries of life. The strong interaction. Covalent bonds in biological molecules. Covalent bonds play a fundamental role in biological systems, providing stability and structure to biomolecules. In biological molecules, such as proteins, nucleic acids, and carbohydrates, covalent bonds are crucial for their formation and function. Proteins. Proteins are composed of long chains of amino acids, linked together by covalent bonds known as peptide bonds. These bonds form between the amino group of one amino acid and the carboxyl group of another, resulting in a polypeptide chain. The sequence of amino acids and the folding of the protein are determined by the strong covalent bonds within the peptide backbone. Covalent bonds, such as disulfide bonds, also contribute to the stabilization of protein structures. Nucleic acids. Nucleic acids, including DNA and RNA, contain the genetic information necessary for life. The backbone of DNA and RNA is composed of a sugar phosphate backbone, where phosphodiester bonds connect the sugar moieties. These covalent bonds create a stable framework for the genetic code, ensuring the faithful transmission of information during replication and transcription. Carbohydrates. Carbohydrates, also known as sugars, are vital energy sources and structural components in biological systems. Covalent bonds, such as glycosidic bonds, link sugar monomers together to form polysaccharides. These bonds provide stability to complex carbohydrates, such as cellulose and glycogen, enabling their storage and structural functions. The weak interaction. Intermolecular forces. While covalent bonds dominate the intramolecular realm, weak interactions come into play when considering intermolecular forces, the forces that exist between molecules rather than within them. Intermolecular forces are responsible for phenomena such as boiling points, melting points, and solubility. The weak interaction, non-covalent bonds in biological systems. While covalent bonds provide stability and structure, weak non-covalent interactions govern many biological processes, allowing for flexibility, recognition, and regulation. Hydrogen bonding. Hydrogen bonding plays a critical role in biological systems. It occurs between hydrogen atoms and highly electronegative atoms, such as oxygen or nitrogen. Hydrogen bonds contribute to the specific pairing of DNA strands, stabilize protein structures, and enable the recognition and binding of molecules such as enzymes and substrates. Van der Waals forces. Van der Waals forces, specifically London dispersion forces, are weak attractions that arise from temporary fluctuations in electron distribution. These forces contribute to the folding of proteins into the compact structures and the interactions between hydrophobic regions in biomolecules. They also play a role in the binding of small molecules, such as drugs, to target proteins. Ionic interactions. Ionic interactions occur between positively and negatively charged atoms or molecules. In biological systems, these interactions are important for maintaining the stability of proteins and nucleic acids. For example, the binding of metal ions to proteins can influence their structure and function, and the interaction between oppositely charged regions in proteins can contribute to their stability. For detailed study, please visit the website biologymum.com and follow the social media profiles. Links are in the description.